Hello everyone. So we take on from where we left off. The last time we were working on adsorption, and what we are going to start on is to look at how to determine KD values through experiments in the lab. Now KD can be determined either by performing batch experiments or using flow-through reactor experiments, and I'll explain that in a little bit. In case of your batch experiments, what one does is that you take your sediments and the solution containing the ion that you are interested in determining the KD for. So for example, in our case, we are interested in looking at the KD for phosphate. So we take a solution of phosphate of a known concentration in different vials. So in this case, we have 4 vials with 4 increasing concentration of phosphate in them. It's 5 millimolar, 10 millimolar, 15 and 20 millimolar. Within the test tubes, we add sediments of known quantity, preferably same weight in all those 4 test tubes. It can be clay, it can be sand, or whichever material you are in which your solution needs to be in contact with. Once you have that, you let the solution equilibrate with your sediments over time, usually within 24 hours. If the reaction is fast enough, this reaction will be in equilibrium, which means most of the phosphate that was supposed to be adsorbed onto your sediments would have adsorbed. At that stage, you are going to take a sample out of each vial and remeasure the phosphate concentration. Now the difference between the remeasured phosphate concentration and the original concentration is the amount of phosphate that has been adsorbed onto the sediment particles. So that gives you the concentration of adsorbed fraction, and you have the solution fraction already that you had the input in the solution. So those two concentrations are then plotted on the curve like we had discussed previously, C adsorbed versus C concentration in solution, and you will get a plot something like this. Depending on what kind of behavior ion is showing, you may have a linear behavior, or the your Langmuir type of behavior, or a Freundlich type of behavior. Once you have that curve, you should be able to determine the KD with the mathematical formulation that we had discussed earlier. So in this case, suppose if you had a linear relationship, then the slope of the linear curve would give you the value of your KD. Now, these batch experiments, they are easy to perform and they are fairly quick to perform. So that's the advantage of these batch experiments. But there's also a problem with these batch experiments, because once you add the solution into the and mix it with your soils or sediments, there are byproducts that are being produced within the vial because of reactions taking place, and those products of those bioreactions can actually compete with the adsorption of phosphate. So you may not have a adsorption coefficient that is truly reflective of the adsorption. So because of that, then there's another method through which you can determine your KD values, and that is through flow-through experiments. As the name suggests, in this case, instead of solution being in contact with soil all the time, in this case the solution is flowing through the porous matrix. So you take your sediments into a reactor of interest, so in our case it will be phosphate, and what you are doing is you are pumping this phosphate solution through the reactor, and on the other side of the reactor you are collecting your samples after every 1 hour or 2 hours, whatever the time may be. And you will keep on collecting these samples till you reach the concentration that is at almost a steady state. Now, in order to determine the KD value, along with your ion of phosphate, or that is the ion of interest, you also need to add a non-reactive tracer. For example, bromide. Now the purpose of this non-reactive tracer is that it basically is there to understand how the flow is happening and the transport of your ion that is purely related to the flow of solution. Because what you want to do is you want to separate that out from the ion concentrations that are affected by adsorption. So your concentrations in these vials are affected by the rate of flow of the solution and the rate of adsorption onto the particles. Once you have these samples, you will analyze for the phosphate concentration and the bromide concentration, and you plot them in terms of what we call the breakthrough curves. So in this case, you're measuring the concentration, output concentration in your vials that you are collecting samples, and C0 is the input solution concentration. So in this case, say for example, you started with phosphate concentration of 5 millimole. So that will be your C0, and in the output, after every hour, you are measuring what those concentrations are going to be. Same thing is in case of bromide, you are measuring the bromide concentration. So you start with 0 concentration in the output, because at that time it's only water that is flowing through. The solution hasn't come out through. Once after a certain time the solution comes out, then you start measuring these concentrations of phosphate and bromide in your output solution. So you plot that over time. So you plot C out over C0 versus time, and you will get these two curves. One is for your ion that is being adsorbed and one for the ion that is non-reactive. Obviously the non-reactive ion will come out first because it is not getting involved in any other reaction, and the response in the reactive ion will be delayed because that ion is being adsorbed onto your sediments. So once you have these two curves, what you want to find out is the half time of the breakthrough curve. So breakthrough, one is basically your concentration in the output is equal to your input concentration. So half of that, which is 0.5, you take that point and you are looking at the half time of the non-reactive tracer and the reactive tracer for that particular breakthrough curve. Once you have those two times, you can calculate your adsorption coefficient. So this is your half time for reactive tracer divided by the half time for non-reactive tracer, which is equal to 1 plus k. Now this k is an adsorption coefficient, but it's not same as kd. This is a dimensionless adsorption coefficient. It is related, so it is dimensionless adsorption coefficient, which is related to kd by this formula. So your non-dimensional adsorption coefficient is equal to rho multiplied by 1 minus phi divided by phi multiplied by kd, where rho is your density of the sediments and phi is the porosity of your sediments. And so this is the sediment that is inside your reactor. So once you have those values, you can calculate your kd value from there.